Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Accelerating Biologics Development to Medicines, Key Drivers of Success. My name is Sonia Hunt, and it's my pleasure to be your X-Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout this presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Aragen Bioscience, who developed the content for this presentation. Aragen Bioscience, Inc., a subsidiary of Aragen Life Science, is focused on accelerating antibody and other biologic product development. Their integrated preclinical research offerings include antibody discovery, recombinant protein production and purification, stable cell line development, as well as cell-based assays and in vivo models of disease. Their team has been chosen for their scientific experience and diversity. They are located in San Francisco Bay Area, California. Now it's my pleasure to introduce you to your speaker, and she is Dr. Sufia Karim. She is the Director of Cell Line Development and Late-Stage R&D at Aragen Bioscience, Inc. She has more than 18 years experience in the biotech industry, including leading cell line development, cell culture, process development and optimization, and successful tech transfer. She earned her MD from Sir Salimullah Medical College in Bangladesh and her MS in molecular and microbiology from San Jose State University. Now it's my pleasure to pass over the mic and the controls over to Sufia. So Sufia, when you're ready, you may begin.
Thank you very much, Sonia, for the nice introduction. And thank you, everybody. Good morning. This is Sufia Karim. I'm the Director of the Cell Line Development. Aragen Bioscience is a contract research organization. We are in the industry since 1993 and located in Morgan Hill, California. We have occupying the two large facilities, one for the PCT business unit, which is headed by Dr. Malavika Ghosh, who is a renowned leader in the field of animal model. Our PCT department is occupying 15,000 square feet building for the rodent vivarium and in vivo laboratories. It has 3 test suites, BSL-2, and 5 wet labs for the analytical and ex vivo in vivo procedures. It is USDA registered, OLO assurance, and LAC accredited. The other facility, the Biologics unit, which is headed by Dr. Stephen Lang. Unfortunately, he cannot be here today because of some family issue. These biologic organizations cover the antibody discovery, reagent preparations, analytics, formulation, and cell line development. We have 28,000 square feet building, and we have 6 CLD suites, 2 upstream process suite, and a dedicated microbial lab. Just beside to that, to the adjacent molecular biology, hybridoma, downstream processing, and analytical lab. And now, recently, we are in the process of building a GMP manufacturing facility, which will have 2 500-liter sub for production of the drug bulk substance. Throughout our 28 years in the industry, we have experience in all biological formats and numerous expression systems. This institutional knowledge is valuable. As you know, the recent molecules are more complex than in the past. Today, I'm going to cover the topic from the industry and our work to accelerate the molecules to medicine. So trying to accelerate the process, we have to think about the product life cycle from discovery, development, and commercialization. To accelerate the discovery and the development is really risky because this is complicated, time-consuming, expensive, and also already in a high-risk endeavor. As you know that more than 90% of the drugs never make it to the market. So remember that our job here is to make the complicated science into a robust, consistent, cost-effective, clinical and commercial supply of process. But with the biological system, it is not always predictable. The product life cycle, that is from the discovery, development to commercialization, no one person knows everything necessary to carry this molecule from the discovery to market. Therefore, it takes a community to have all this work together and trying to accelerate so that we don't go through the problem and we can reach the client as soon as possible. Sorry about that. In this figure in the middle portion, this is taken from a paper published by Stephen Paul. It is the economic analysis of the drug development process. He is trying to highlight the different stages of the drug when it comes from the preclinical stage to phase 1, phase 2, phase 3, and ultimately submission for the NDA. So in different phases, where is the main problem happening? And as you can see, as I mentioned, 90%, more than 90% doesn't make it into the market. According to Stephen Paul's hypothesis, the most of the failure happened during the phase 1, about like 36% and 56% at phase 2. So this big area of failure for the drug to come into the market, he called this one as valley of death. This is the economic analysis. He is saying the challenge is the failure in phase 1 and phase 2, that is the valley of death. We need to focus on this portion, and he given the solution that we should avoid this valley of the death by investing more in the upfront, in the preclinical stage, where we can invest more and then we learn about the process, learn about the molecule before, and also make a robust process before we submission for the IND. That will avoid this full loss in the later portion, which is very expensive. As you can see, for an example, for the phase 1, one of the run in the phase 1, it takes about 3 to 5 million. So instead of wasting the time and failure in the phase 1 and phase 2, it's better to invest less money, which is less costly, in preclinical stage, learn about the molecule, narrow down the molecule, which is important, and then make a robust process and apply for IND. So during this stage, Aragen and most of the industry taking his advice to heart to improve the preclinical stage. We think the preclinical stage as a critical junction between the discovery and the development. So where this in the preclinical stage, we generate the critical product information, and then huge efficiencies can be built around this process. This will be the topic for the rest of my talk. As we start discussing about the acceleration of the program, we need to understand the best way to mitigate the risk is understanding the product. While there are options available, like during the lead optimization and early preclinical stage, where there are still options available and we can choose the best candidate. This is the critical bridge between the discovery and development. In discovery, we are designing and learning about the product, and in development, we are developing the process to produce the product. Recently, the advancement in the CMO business over the last 10 years has greatly improved our ability to accelerate the program without taking any significant risks. Those advantages are the platform process knowledge recycling, advanced analyticals, and developability to drive the real-time decisions, the process levers to control the critical quality attributes, and outsourcing to leverage the external expertise and focus on core competencies. So I will talk about this more detail from the rest of my talk. But bottom line is the company that leverages the intelligent R&D that is focusing on understanding of the product and strong collaborations with the CRO or CDMO collaborations like Aragen, who has extensive process knowledge, can successfully accelerate the program without addition of the risk and build the competitive advantage. But we must understand when we are accelerating the process, we are working with a limited knowledge. Limited knowledge about starting material production, limited knowledge about the GLP tox and phase 1 production, limited formulation stability, and limited cell comparability study between the RCB and MCB. Since every biological candidates are unique, when biosimilar, even with the biosimilar, the companies are behoves to identify the knowledge gap early. So now, when we are trying to fill the knowledge gap early, we are accelerating and we are trying to accelerate the project. It is most important to focus the limited resource and time on the critical attributes. So, in a few years back, we started the QBD program, that is the Quality by Design program, where we process on the focus on the product knowledge in the form of CQas, that is the critical quality attributes, and the process knowledge in the form of CPPs, that is the critical process parameters. As we know, the critical quality attributes, which are really related to the safety and efficacy of the drug product, which includes like identity, purity, quality, potency, and strength. And CPPs always influences the CQs. The caveat is this: the understanding of the product CQs means that we must have the early knowledge of the mechanism of action of the drug, the potential for immunogenicity, and how the CMC process will impact the mechanism of action. The most product, we don't have this kind of knowledge, especially for a complex molecule. For example, like Humira is a simple monospecific antibody, binds with one cytokines and only have a one mechanism of action. Whereas recently, most of the molecules like bispecific, which can have 2 or more mechanisms of actions. Failure to understand the CQs and CPPs early, we have to go through that lot of risk and the program will be failed. So key to success is leveraging the early and exhaustive analytics, previous knowledge from the platform, and the levers to mitigate the risk, and overall the community of experts. Sonia, can we have the poll question?
Certainly, Sophia. I'm going to launch a poll question right now. So audience members, this is done in real time, so I'll be able to share the results with you once I close the polls. I'll give you about 40 seconds to complete the simple poll question. And the question that we have for you today is, how important is speed in your overall decision while picking a CRO CDMO? So please go ahead and select from the below. And you have 4 options there: somewhat, not quite, highest priority, not important. So please go ahead and cast your votes. Okay, that looks like it's the majority of our audience. There's still a few more members out there who have not cast your votes. Can you go ahead and do so now, please? Greatly appreciated. Perfect. Thank you, everyone, for participating in our first poll question. Now, let's take a look at the results. Closing the polls. Okay, so here's the result of our first poll question. We have 28% of our audience members are saying somewhat, 4% that said not quite, 60% of our audience members are saying highest priority, and then 8% saying not important. So thank you, everyone, for participating in poll question number 1. Now, Sophia, before you continue, what are your thoughts about the results? Are you surprised?
No, this is very encouraging. Yes, we have seen that like all the clients we are getting, they're all looking forward, like how fast we can go, how
Mm-hmm.
fast you can take it from the cell line development all the way to the R&D submission. So that is the highest priority. And from recently, we all go through the pandemic, and we have seen that the every other small company, big company, everybody is fighting to find out the vaccine for
Yeah.
COVID, the
Yeah.
treatment for COVID. So definitely that is the highest priority. Who can bring out fastest, they will win the process, and we can help the patient as soon as possible. So
Okay.
it is not surprising. This is really, really real data.
Okay. Thanks.
So now, as you know, that we are talking about to move the process faster. So as I talked earlier, that we need to involve in the fast in the preclinical stages and how we can make it faster through exhaustive analytics. Analytics is our eyes and ears. No amount of analytic testing will improve the product quality once you select the candidate and the process is locked. So we have to put the analytic development way early into the stage. Usually the most analytics happening during the process development in CMC stage, but now as we wanted to make it faster and effective, we are pulling it in front. That use of the tools and methods for the CMC analytic development during the end of the lead optimization and throughout the preclinical to establish, to have the consistent data from different stages, which is most important and important for the product, and identify its purity, quality, potency. So basically the critical quality attributes. Use this method preferred by CMC analytical development to screen the candidate from the very early stage like lead optimization when many of the candidates are here. De-risking is possible when you have the option. Use the analytic process that will help to uncover us the known and unknown liabilities, such as stress studies. We will talk about that in a few minutes. And finally, we wanted to take a holistic approach during doing more analytical work earlier to create the marketable drug. When Aragen is talking to the client about discussing the acceleration, we try to think about the proof of value rather than proof of concept. Because the proof of value consider both the patients and the business need when designing a product. So there is no sense of a company to make a drug which the market doesn't want it, right? So the analytic development is a great tool for us, as I mentioned. It is one of the examples of early analytic development. It's published by Genentech, especially Bill Sawyer and Amy Shen in 2020, demonstrating that the power of rapid mass spectrometry. This is a very high throughput sample preparation, mass spectrometric analysis, and automated deconvolution process that generate a large volume of accurate mass in a shorter time. Basically, they are using a Kingfisher sample prep instrument with the use of the magnetic bead to isolate the maps from the cell culture fluid, and when they are injected to the machine. And it analyzes by mass spectrometry to produce the molecular weight data. The power of the mass spectrometry, as you can see, it is very important in the early stage. Suppose like this is a bispecific molecule we are targeting for, and the outcome can be like a lot of impurities along with it, more than 10 plus impurities. But we need to identify the best one, which is producing our target of interest, the best one, right? So with use of this high throughput rapid mass spectrometry, the analytical system demonstrating the differentiating the quality and like a knob-in hole process in this bispecific thing. The high throughput analytic can be used during the CLD work to quickly identify the best clone based on the product quality. It's not only the titer, the quality is more important, but in the bispecific, we are having a lot of byproduct impurities. When we go through this high throughput mass spectrometry, easily we can identify which clone is producing the mostly desirable product. And then we can use in the next stage to take those clones to move forward. Ultimately, the top clone will be selected based on the quality, not for the title. This is a powerful example how the high throughput analytic method used in the preclinical stage rather than in the CMC pharma development, which can be identifying the candidate in the CLD and optimum product quality and mitigation of the risk strategy so that we can move to the clinic faster with a better title and the quality. Another analytical advancement recently happening is the developability step. Early analysis is developability. It is also called the molecular assessment. We built our developability package. That is work done by Steve's previous group in J&J, which has become the industry standard now. This slide shows the high level of developability process built by Aragen to identify the potential CQs. As you can see that it starts in the in silico release, where we are checking the biophysical analysis, post-translational modification, degradation, liabilities, and also looking at the cross-functional motif. And based on that, based on the molecule, the release testings are given, like which test is important for this particular molecule. After that, with different kind of stressing the molecule, depending upon the molecules and the client's interest, we can do the oxidation, deamination, different pH to check the aggregation and fragmentation, and also like during formulation, concentrating the material and see whether over a time it is changing the quality or mechanism of action or efficacy. So that helps us the insight. It's a structure-function relationship and the critical quality attributes to meet the patient's need. It is done very early with multiple clinical candidates to demonstrate a method to accelerate the program by eliminating the poor candidate, by uncovering the unknown liabilities and building the mitigation strategy before the CMC pharmacy development, analytic development begins. So after this analytical advancement, we are moving towards the next stage, how to accelerate that. Before going to that, Sonia, do you have any other poll question?
Yes, I'm going to launch our second poll question now, Sufia. And here it is, audience members. Okay, just like before, it's done in real time. I'll give you 40 seconds to complete the simple poll question. And the question is, what value do you place on early risk assessment in your journey from sequence to the clinic? And then once again, there's 4 options below to choose from. Please go ahead and select them now. Somewhat valuable, not quite valuable, highest priority, or not important. So please go ahead and make your selection. Okay, that looks like it's the majority of everyone. I'll just give you a few more seconds and then I will close the polls and then we'll get the thoughts on the results with Sufia. Okay, thank you everyone for participating in poll question number 2. I'm now going to close the polls. Okay, so here are the results. We have 11% are saying somewhat valuable, 0% are saying not quite valuable, 89% of our audience members are saying highest priority, and then 0% saying not important. So thank you everyone for participating in poll question number 2. Sufia, are you surprised by the results of that poll question?
No.
No.
Oh, this is very good. Yes. That is the way we are moving forward. Like we really need to look at, just like I was talking in the very beginning, Steve Paul's analysis, right? So everybody wants to give the risk, like we are seeing that phase 1 and phase 2 is the risky process, to bring it up front and check it out, the molecule in the beginning before it goes to the clinic. This is very normal result. I was expecting.
Okay, perfect. Thank you.
Okay, so now I see talk about the analytics. So we understand about bring all this together in a lean R&D in order to make a better decision faster. Now we have the examples of the tools to use in the early stage to de-risk and accelerate the program. We can now turn how to shorten the timeline. Because the tools, we know that with QBD, we know the product quality attributes, product process, critical process parameters. We know the upfront load for the analytics gives the more information regarding the molecules. Now we can focus on how to make it faster with minimum risk. There is regulatory business risk and also safety for the drug and patients. To make for the accelerating the process, now we can start talking about the parallel processing, how to make it faster. So in the upper portion of this slide, you can see this is the conventional way of the CMC process, where one step finished and the next step starts, just like we start from the lead optimization, then clinical. We select the candidate, go through the CLD, make the cell line, make RCB. Then from the RCB, we are doing the process development in bioreactor stability. And at the same time, with the RCB, people start doing the phase 1 process development, and then that is used for the tox run and parallelly going to MCB and ultimately go to the phase 1 IND preparation and submission. All the way once finishing and another studying, it takes from start of the CLD work all the way to IND submission is about 20 months. It's a long time. You don't have any clue what is going at the end, how the data turn out. So now we are using our earlier investment in analytics and earlier investment in the developability study to make it faster, do the parallel processing. And the lower portion here, you can see that we moved everything towards the left. So making the whole timeline shorter from 20 months to around 12 months or 15 months, because we are starting the process development and the downstream development, not waiting till the RCB is met. We are starting when in the middle of the cell line development, where it is not from the RCB, the midterm product from before the RCB. So it will be quite a good representation ultimately in the bioreactor, the main process log. And this process will be used for the tox study. So it cut down the tox time and moving it faster. Instead of using the RCB, we are starting before that and cutting the timeline there. So the functional, now we have the framework. We start the parallel processing, shortening the timeline, and that will help us to go to the clinic way earlier. So how we are using and making sure that we can move that fast. Main thing here, we can see that platform process knowledge, that is the key for the key accelerator for us, because we don't have to recreate it over and over again for new program. We have standardized the media, feed, and the reagent for the downstreams. Here at Aragen, we always use the commercially available media and the feed, because the last 5 to 10 years, there is a significant improvement in the quality of the media. The media now make the process, this media, we know which media is best for one particular cell line, typical cell line to use. We have the process knowledge on that. And we can easily transfer this process to the CDMO because they have the commercial media available and they can restock their process. We use our previous knowledge which we did in the past. We can do all this troubleshooting. By using our past experience with the expression platform, we can predict the timeline also in the IND and plan for the areas to accelerate. So Now we talk about accelerating with our knowledge from the platform development. And also here is one of the example like Genentech is using in the bottom graphic, bottom picture. You can see I took this one from the Genentech. They are making it much faster there. They are using the transient material here. This transient material, the strategy of the using of the transient material for the GLP dogs and also not waiting for the RCB even before the CLD starting. So showing here the case study is helping us to show that this transient work and the CLD work, when the CLD is completed, the analytics and the formulation and the downstream process is already having some idea to move to the next stage. So approach is to expand our process in the phase 1 to introduce the greatest product quality and diversity. Finally, to really accelerate our program, it requires a business risk in the form of cost and to support for the parallel activities. The key to the negotiation with the project leader to understand the risk and opportunities. It starts with the downstream processing, formulation, and analytical method development from the transient material, generate the more representative material for the CLD and resupply the downstream material in the form of analytics. We start the conversation early with the CDMOs and stockpile the necessary material to get into their schedule. Begin the phase 1 process development when the platform fit exercise and use the levers to control the product quality attributes. And finally, we have to negotiate the CDMOs to use the interim release of the MCBs, which can be used in the phase 1 downstream process development. This type of acceleration is the best done for the monoclonal antibodies, but for the complicated material, we have to develop a couple of the process like this, and then we can apply this knowledge in the different formats too. So working closely with the project leaders in the CDMO partners early before the beginning of the process is essential to make them understand that how it is important, how it is like everybody is saying that we need faster and faster. To make it faster, to remove the risk mitigation strategy, we have to start very early in the process to make them understand why we have to put a lot of analytics in the front, have to do parallel processing to go and mitigate the risk and go with not jeopardizing the quality and have a regulatory success. So after all this talk, I hope everyone appreciated that our learning from the last 10 years in the biologics manufacturing that can be used to any type of program. However, it takes a community to drive and shift from the conservative CMC development to lean thinking with the involvement of the process knowledge and expertise and advancement and craft to have a successful plan to accelerate the process. So in conclusion, the acceleration begins in the form of discovery and development and ultimately the market. It requires a community of experienced leaders, that is CRO and CDMOs, exhaustive conversation and understanding of the biologic candidates, the risk, the clinical plan, and the potential for the patient's need. Early identification of the product qualities to focus on the analytical work, extra work, and the risk in the form of parallel activity to accelerate. Understanding the downstream work in order to facilitate their work, and continuous update the risk assessment and plan how we can learn more. So In our goal in the industry, especially in Aragen, we focused on increasing the speed to cleaning by increasing the efficiency and also predictability while decreasing the cost and technical, clinical, and regulatory business risks. To avoid all this, we need to avoid the discovering the, we have to avoid, identify the liabilities and the product process knowledge before it is too late. Thank you. If you have any questions, Sonia, we can go to the questions. Thank you very much. Thank you, everybody, for listening to my talk.
Well, thank you very much, Sufia, for that very engaging presentation. I hope everyone enjoyed it. Before we start the Q&A portion of the webinar, we have one last poll question. I'm launching it now. Okay, would you be comfortable performing your cell line development and phase 1 manufacturing at different sites or with different companies? And then there's 4 options below, just like before: somewhat comfortable, not quite comfortable, absolutely okay, will not consider. So please go ahead, cast your votes, and then I will close the polls, share the results, and then we'll get right into the Q&A. Okay, that looks like it's the majority of everyone. And as usual, I'll keep it open for a few more seconds. For those of you who have not cast your vote, you simply just have to click on one of the above or below. Okay, perfect. Thank you everyone for participating. Now I'm going to close the polls and let's take a look at the results. Okay, so we have in our audience 21% are saying somewhat comfortable, 26% are saying not quite comfortable. We have 47% of our audience members are saying absolutely okay, and 5% will say said will not consider. So thank you everyone for participating in poll question and our final poll question for today. And now it's my pleasure to open up the Q&A portion of the webinar. For that, we'll put on our webcam. So Sufia, you can put on yours. And remember, audience members, you can continue to send in your questions right now by using that questions window located on the right-hand side of your screen. There's Sufia, and we'll try to attend to your questions during the time that we have together. Sufia, while you were speaking, I did receive a bunch of questions from our audience members, so I'm going to start with those until more come in. And this is our first question. This audience member is asking, would you recommend testing multiple clinical candidates in developability and or early stages of CLD?
Yes, we highly recommended taking multiple candidates in the developability package, even in the CLD, if you have the bandwidth. Even if you don't have multiple candidates, it is a time. You can check the opportunities and just modify your effector functions and see whether it is, if you enhance it or reduce it, whether it is affecting the mechanism of action during the cell line development.
Okay, thank you so much, Sufia. Let's go to our next question. Okay, this audience member is asking, how far do you think we can take pools, non-clonal cells for production? It's interesting.
Yeah. So recently, one of the work done by Boehringer Ingelheim, they are taking the non-clonal pool cells up to 2,000 liter scale. And they have proven that it is phenotypically, genetically, and the quality consistency up to 2,000 liter scale, which is pretty good. And they are trying to use this material for the first in human use. So nowadays, from the clonal to pool to go to further tox run is becoming more popular, and with our advancement with the analytical development, it is possible, and we are looking more and more on that.
Okay, perfect. Thank you, Sufia. Let's go to our next question. All right, here it is. For challenging to express molecules, do you have a strategy to determine the best expression platform for CHO or CLD?
Yes, we at Aragen, we offer 3 different platforms, CLD platform, and it is a cost-effective approach for the beginning. We usually encourage clients to start with all 3 different platforms up to a certain stage, and then based on the data, you decide which one is the best one for your candidate, and then move on with the one for the full CLD project. And definitely 3 different platforms comes with different licensing cost.
Okay, all right, thank you. All right, here's our next question. Okay, what is your current timeline from DNA to IND?
We have a good partnership with the CDMO and with a good relationship and with good collaboration from the very start of the program. It is now from DNA to IND is about 15 months.
15 months? Okay, all right,
Yes.
thank you. Let's go to our next question. What is the most important factor to accelerating a program? This looks like it may be our last question.
In my opinion, it is the experienced program leader who can answer our questions and give us the path directly, which is the critical path for moving forward. So pairing up with a good CRO-CMO combinations is the best way to accelerate the program, start working closely from the very beginning of the program and do all the parallel processing, get the analytics ready according to the CMO's availability, their equipment of use, and the molecules need. So definitely it is critical, most critical is the good communications between CDMO and CRO.
Okay, thank you. Let's go to this next question here. We have another one just came in. Why bispecific antibodies result in low titer? How can we improve it?
Yes, so one of my slides I was showing you the analytics, high throughput analytics for the bispecific, because bispecific has a lot of impurities. You don't have the control over them, like which portion will be forming perfectly, right? So we have to identify the best clone which is producing the most desirable format. So if you just look at the titer, then you will be really focused to the other side, not picking up the best clone which is having the best quality titer. So titer is not the main thing to drive the project. It should be the quality to drive the project. That will be a successful path.
Oh, okay. All right. Thank you, Sufia, for that. Okay, that looks like it's all the questions that we're going to answer for today. So I just want to say thank you, everyone, for sending in your questions. Now, if we didn't answer your questions, the team at Aragen will follow up with you after this presentation. And if you have any further questions, any inquiries, please send it to info@aragen.com. They'll be very happy to speak with you. Okay, so thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now give me a few seconds. I'm going to send you a link in your chat box and you'll be able to view the recording of this event with that link and also share that link with your colleagues once they register for the recording as well. So I encourage you to do that. And get even more out of today's presentation by downloading the supporting materials that are available under the Handouts tab. And we have two there for you, Aragen CLD Platform Testing, and then the other one is Aragen CLD Solutions. So please go ahead and check it out. Now, please join us in thanking our speaker, Sufia, for that very insightful presentation and for answering your questions. So thank you, Sufia.
Thank you, Sonia, and thank you everyone for having me in the talk. And if you have any question, as Sonia said, please don't hesitate, contact us. We will get back to you.
For sure. Thank you. Thank you. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Thank you for joining us. Thank you, Sufia. Bye.
Thank you. Bye-bye.